Day 81 of creating a microbial sandbox by adding random stuff and observing it under the microscope every day. Yesterday, we added tomato. Now the sandbox is red, turning into a tomato soup. Let's put it under the microscope. I see some red substances inside Paramecia. Maybe they ate tomato as food. The whole sandbox is actually covered with fungal hyphae. Other microorganisms are using them as shelter. Do you remember this? This is Dileptus. It is a predator that can touch and paralyze other microorganisms before ingesting them. Look at this tragic ciliate. It suddenly burst in the middle of filming. Remember, we put a part of the sandbox in a dark environment a week ago. Let's take a look now. Actually, the microbial population is quite similar to the original one, with a slight difference of having fewer microorganisms, especially bacteria. I think it is because it is kept indoors, unlike the original sandbox, which is outdoors, hotter, and acts as a natural incubator. I bought some fish roe. Let's take a look under the microscope. I can see it is empty inside. After breaking it, the internal fluid flows out. Let's put some into the sandbox. That is all for today. See you tomorrow. Day 82 of creating a microbial sandbox by adding random stuff and observing it under the microscope every day. Later, we'll conduct a stress test on the microorganisms. But first, let's take a look at the sandbox under the microscope. Paramecium now seems to have overtaken Vorticella as the dominant species in the sandbox. Besides them, there are other much smaller microorganisms. They're mostly ciliates, a large and diverse group with many different species. The predator has returned. It's like the microscopic lion of this ecosystem. I also spotted something that resembles Vorticella, but it's at least 100 times smaller. These tiny organisms anchor themselves to surfaces and move rapidly. Some of you asked me to try acid, so let's test it. I have hydrochloric acid, a strong acid. I'll add just a drop near the cover slip. As soon as they came into contact with the acid, the organisms gradually stopped moving and eventually burst. This acid is too strong. It kills everything, even those hiding in organic debris. Now let's add some other stuff. First, toothpaste. I'll test it on a small portion of the sandbox before adding it to the whole jar. Next, turmeric. This is a supplement I used before. Let's add some and see what happens. That's all for today. See you tomorrow. Day 83 of creating a microbial sandbox by adding random stuff and observing it under the microscope every day. I am thinking about whether we should find a day to arrange a live session to do the experiment together. Let's take a look at the sandbox under the microscope. In just this view, I can see 5 different types of microorganisms. Can you count them? All of them have been introduced in the previous videos. These microorganisms really like living inside fungal hyphae. It creates spaces for them to hide and gather. I do not know why they gather and form something like a path, as if they are guiding us somewhere. Some of them anchor themselves onto objects like algal filaments. These small ciliates are blooming in the sandbox. It makes me think of a group of mosquitoes gathering around. Remember we mixed a part of the sandbox with toothpaste? Let's check the result. I do not see any organisms under low magnification. When I zoom in, I can still see a few bacteria swimming. The antimicrobial effect is still very strong. Let's add stuff. A sponge from the kitchen. Since it contains detergent, I want to test it with a portion of the sandbox first. Aloe vera. Personally, I would rather put it on my skin than into the sandbox. That is all for today. Day 84 of creating a microbial sandbox by adding random stuff and observing it under the microscope every day. For those curious, the water bears are still alive in the paradise box, but their numbers haven't really increased. Let's put the sandbox under the microscope. Around the organic debris, there are millions of microbes. There are mainly 2 types. One is larger and moves with a cockroach-like motion. The other is smaller and slowly flows around. Let's zoom in. This is what the smaller one looks like. There are also some other microorganisms that look like they have arms and legs moving around. I think this might be the same type that burst in front of us before. Remember the part of the sandbox we soaked with a sponge? Let's check on that. Once again, most of the microbes seem to have died. Only a few bacteria are slowly swimming. Today, we're adding coffee grinds. Some people say they provide nitrogen. Let's try that, along with a bit of caffeine. That's all for today. See you tomorrow. Day 85 of creating a microbial sandbox by adding random stuff and observing it under the microscope every day. It seems they like coffee grinds. They concentrate around the grinds, especially paramecium. I found a specially shaped paramecium. It looks like a boomerang. It might be a dividing paramecium, but it actually looks a bit different from the ones I have seen before. Could it be a mutant? There are also some small ciliates present. Around other organic debris, there are mainly these small microbes, which require 400 magnification to observe. Rotifers are also living among branches of fungal hyphae. Let's add stuff. Chili. This is Thai chili. I will need more time to find other specific types. Let's test it with a portion of the sandbox and check the result tomorrow. Raw egg. We have not added egg white before, so let's try it now. That is all for today. See you tomorrow. Day 86 of creating a microbial sandbox by adding random stuff and observing it under the microscope every day. This is a low magnification view around 40 times. Let's zoom in to over 100 times. You might wonder why the video looks laggy. That's actually the real motion of these microbes. Weird, right? At over 400 times, you can clearly see the morphology of the microbes. Some even look like nuts. There are also crescent moon-shaped organisms. That's Rhaphidocelus, a genus of algae common in freshwater. It grows quickly. Here's Iolosoma hemprichii again. It's a common worm found in polluted water. Let's add stuff. Remember we mixed part of the sandbox with chili? Let's check it. I can't see any microbes except bacteria. Are microbes really scared of spicy stuff? Let's add just a little bit to the sandbox. Next is spirulina, an algal supplement. Let's dissolve it and observe it under the microscope. These curved green cells are spirulina. The concentration is lower than expected. I'll add some to the sandbox. Now a swab for my phone and computer keyboard. They should be clean since I clean them regularly. That's all for today. See you tomorrow. Day 87 of creating a microbial sandbox by adding random stuff and observing it under the microscope every day. From this day on, I will take both the surface and the bottom of the sandbox to the microscope. Good news, the sandbox is now crowded with paramecia. The bad news is that I hardly see any microbes other than paramecia. Natural selection chooses paramecia to survive. Now I can confirm that the boomerang-like microbes is a dividing paramecium. Anyway, it is a good chance to use the high paramecia density to do some experiment. Previously, we added red 40 to the sandbox. Now we try to add it on the slide. It is funny to see that when the red 40 reached to them, they immediately spread away. It seems they are scared of it. When they are immersed into the dye, they die immediately. But it seems they didn't take up the pigment. Maybe the concentration is too high. Let's do it with more dilution. It seems they can endure diluted pigment. Let's add some into the sandbox. Seed. Can they successfully germinate? That's all for today. See you tomorrow. Day 88 of creating a microbial sandbox by adding random stuff and observing it under the microscope every day. I found a green nematode. It's actually the most beautiful nematode I've ever seen. As usual, paramecia are the major population. I think I need to start thinking about how to increase the diversity. Even so, there are still some other common microbes here, like this paranema, and a lot of shivering bacteria. What an astonishing view. It's a good chance to take experiment requests again. Some of you asked me to dissect a microbe using a needle. Unfortunately, a normal needle is much thicker than a paramecium, so it's basically impossible. One of you also asked me to shake the jar. I shook it for 3 minutes. Magnificently, most microbes are still active. But the bubbles created a microscopic maze. Let's add something now. Activated charcoal. Hopefully it can clean up the toxins in the sandbox. That's all for today. See you tomorrow. Day 89 of creating a microbial sandbox by adding random stuff and observing it under the microscope every day. By the way, the whole sandbox changed from red to yellow. And it seems that the activated charcoal didn't kill the microbes. Look at this. I think these should be a special species of Vorticella. It looks like a creature from Biohazard. Again, there are many paramecia inside. I have prepared a new experiment for them. Let's heat the slide for a few seconds. They immediately stop moving and start to burst. It seems they are very sensitive to heat. Time to add more stuff? Many of you asked me to pump air. I cannot pump air inside, so I decided to add more oxygen tablets instead. Ginger. I try to put it near the slide to test its antimicrobial effect. It seems it is not strong enough to affect the paramecia. That's all for today. See you tomorrow. Day 90 of creating a microbial sandbox by adding random stuff and observing it under the microscope every day. As usual, I will look at both the bottom and the surface of the sandbox at the same time. Our old friend paramecia again. I won't introduce it again. A specially looking rotifer. It is a species we have never seen before, called squatnella. Its head is round like a fan. A hypotric, a common ciliate found in dirty fresh water. And also, there are other unknown ciliates with different shapes. One of you asked me to look into the gunk on the walls. Let's check it. Interestingly, you can even barely see some things moving with the naked eye. It means they are so large and so many. Those moving things are paramecia again. It is interesting to know that even though they are single-celled microorganisms, they can still be observed with the naked eye. Okay, let's add stuff. A lime? Add some vitamin C to them? I know we added a cigarette before. This time, let's mix tobacco with part of the sandbox and check the result tomorrow. That's all for today. See you tomorrow.